All right, today we're going to be talking about bryophytes, the first land plants. So obviously they're pretty small, right? They don't have true roots, they have rhizoids, and they can't get very tall. So we're going to look at several divisions. The Hepatophyta, which is the leafy liverworts, and the thallus liverworts. So we looked at a thallus liverwort in there. I haven't been able to find any leafy liverworts. And then division Anthopterophyta, which are the hornworts, which you guys looked at. And then division Bryophyta, which you also looked at. And interestingly, what I've been noticing is that the mosses are one of the first plants to come out in the spring. And they're already developing their sporophyte. And I can't seem to find anywhere exactly when fertilization happens. I don't know if it happens at the end of the growing season or maybe it happens under the snow. I have no idea. So if you guys find something about that, please let me know because it's driving me crazy. All right, so what are the bryophytes? So we call all of these things bryophytes because they're short. If we look at our phylogeny, we can see that the green algae is found right here. It's kind of an offshoot. It's an off group or an out group in this phylogeny. And notice that these synapomorphies here, we have the cuticle, it's terrestrial, has stomata or air pores, and rhizoids on the gametophytes. So can't remember what those other two are. Hold on a second. It's blocking me. Oh yeah, alternation of generations and the embryo. Okay. So before that, what we have is just a very simple type of plant. And remember it had chlorophyll A and B in it. So because this has chlorophyll A and B, we know that it had to be related to these. And then we can go all the way up to the vascular plants, right? So all of these are considered to be non-vascular because they're missing those tracheids. Okay, so all of them, they lack vascular tissue, and they have what's called homosperous alternation of generations. So they don't have small and big spores. And big and small spores, as we go through this, you're going to see that that was really important for actually forming plants with pollen and seeds, essentially. So they have a dominant gametophyte. So when we were looking at our mosses, what you saw was that most of that moss was green, right? The only part that was basically the sporophyte was just that little stick growing off of the top of it. So they do sexually reproduce and they produce spores. So mosses, they basically distribute their spores by water, essentially, because they do have swimming sperm. And then, of course, they have rhizoids, which help anchor them to the substrate, essentially. And they do increase surface area to volume ratio, which can have some absorptive effects. But you're not going to find mosses in very dry environments, right? Because they just can't tolerate those conditions. They can't reproduce without water. So that's why I got so excited about them moving here, because I've never seen so many. All right, so let's talk about homosperous alternation generation. So we have a gametophyte, which in this case is the little leafy part. Don't you like my little leafy part? It's just like a box with dots. And then through mitosis, what this is going to be happening sort of at the tips of those little tiny, they're not true leaves, so they're just kind of little tiny reproductive structures that through mitosis are going to produce the archegonium and the antheridium. So which one, because we talked about this last time, which one produces sperm? So the archegonium or the antheridium? Antheridium, right? So by default, which one produces the egg? Yeah, the archegonium. And then those produce sperm and egg through mitosis, It has to be mitosis, right? Because it's already haploid. And then fertilization occurs, and that's when you produce a diploid zygote. That diploid zygote is going to grow into the sporophyte, which is actually what we were seeing growing off the top of those little tiny reproductive structures. And then through mitosis, they produce a sporangium. Okay, so again, that's that little golf club that's growing off the top. And then this produces spores, but those spores, right, now we have mitosis occurring. So what process is happening right here? Yeah, meiosis. Because you just went from a diploid to a haploid organism. All right, so let's look at the general structure of the Archegonia and Atheridia. So again, we're looking at the tips of those little leaf-like structures. Sometimes they're called phyllings. And inside of them, we have an archegonium. The archegonium is always going to look like a cup with a tail. Okay? And they have these different parts. So they have the stalk attaching it, the venter, which is kind of an opening. We have the egg on the inside, and then we have what are called neck canal cells. In the males, there's a stalk, there's spermatogenesis or spermatogenous tissue, and a sterile jacket layer. So spermatogenous tissue, obviously that's producing what? Sperm. Okay. So in which generation of the life cycle are these gametangia found? Are they in the diploid or the haploid part? Yes, so they are in the haploid part of that life cycle. So again, those gametangia are going to be producing eggs and sperm. But they do it by what process? Mitosis or meiosis again? Mitosis. Yeah.
It's already haploid.
It's already haploid, so mitosis. Okay, so let's talk about the major geologic periods. So we have the Cambrian, Ordovician, Silurian, Devonian, Carboniferous, Permian, Triassic, Jurassic, and Cretaceous. So basically, how do you remember this? Camels often sit down carefully, perhaps their joints creak. Okay. Yes.
That goes off. Sorry about that.
That's all right. And for our absent person, Seth, Mark's phone went off, so that was kind of funny. I don't know if you could hear it. All right. So these are the important geologic periods that we're going to be talking about. Okay. So we think that bryophytes evolved during the Ordovician period, so 450 million years ago, right? It's really not that long ago in the scheme of things. So life has mostly existed as just single-celled organisms until this point. So the Earth was covered in shallow seas. So you can see that the continents are different, right? And there's a lot of very shallow places on the land. And we had a mild and humid climate. So obviously, mild and humid is good for the mosses because that allows liquid water to bathe them and allows reproduction to occur. All right, so let's look at a couple of forms of liverwort. So you guys looked at a thallus. Okay, so we have a thallus over there. And here we have leafy liverwort. So you have phyllids here, which are those primitive leaves. So we also call the little green structures in mosses phyllids. But guess what? They're only one cell layer thick. So I've already told you that they're not true leaves, right? I forgot. There was a question in here I was going to ask you. So what do true leaves have? Okay, if this does not have a vascular system of any kind, then what do the leaves lack? Venation, yeah, but you're not wrong, okay? So basically, they're to be a true leaf, you need at least one vascular tube going through it, and it has to have tracheids in it. Okay, so here's some leafy liverworts. You find them in moist habitats. Actually, I think I do have a leafy liverwort in that container. I think you have to look below the mosses. Okay, here's the thallose liverwort marcantia. So if you looked at my liverworts, there's maybe a couple places kind of towards the tip of that leaf where it looks like there's something forming, but not quite yet. So they're very cute structures. They have these little plantlets on the inside, and they get catapulted out when a drop of water hits them. So we call them gemmae, the propagules, which are the little leaflets, and we call that a gemmicup, where the arrow's pointing. Okay, so here's a cross section, and I think we looked at this in Bio 110. Yeah, I couldn't remember if it was 110 or 111. Oh yeah, so there is a video
There's
that.
very good videos on this stuff.
Yeah, I'll have to play one. Okay, they have this really weird structure though. They have oil bodies in their cells. Yeah.
Now, like, what kind of oil is it? Like, is it really oil, or is it just like fat?
I just missed my mouth.
Oh, sorry about that.
I was drinking coffee. That's for Seth. Okay, so we didn't talk about the doctrine of signatures, so don't worry about that right now. Okay, but I'm not sure what type of oil this is specifically, but it isn't oil. It is not a solid. Okay, yeah. So people used to give people liverworts if they had liver problems. So this is back when people thought, well, if it looks like a body part, it must treat a body part, right? So that's why they call it hepatophyta. So hep, anytime you see that, that has to do with your liver. Okay, so let's look at the life cycle of Marcantia. So if we start here with the Archegonia and the Antheridia, and I'm really hoping that these guys start to sexually reproduce so you can see what they look like in person. So you have the Archegonia. It is visited by that sperm through a raindrop. So once that raindrop hits that the antheridial head, the sperm fly out. Okay, and then they're going to fertilize the Archegonia. But they also have gemmicups, and since the gemmicups do not have gametes involved, what sort of reproduction is that? Is it asexual or sexual?
Asexual.
Yeah, so it is asexual. Okay. So here you have Archegoniophores and Antheridiophores. So what that means, anytime you see phore in Latin, it means carrying. Okay? So these are the structures that are carrying these spore-producing structures. So when water hits those, they're actually going to release spores, and they're going to run or go into the young gametophytes. Okay, so I kind of did this backwards, but you get the idea. So we have sperm and spores produced here. So let's look at the process of fertilization. It is water dependent, and it occurs in the Archegonia. So the sperm need to get into the Archegonia in order to fertilize those egg cells contained within. Okay, once fertilization happens, you have a zygote, which is diploid, an embryo, which starts to develop, and then finally you're going to have sporogenous tissue, which is diploid, and a young sporophyte. And here you have an enlarged archegonium, which we often call the calyptra, which is a covering structure. Okay. So this sporophyte, if it's producing spores through mitosis, wait, no. Okay, so if it's producing haploid spores, then what does that sporophyte have to do to produce those haploid spores? Mitosis or meiosis? Meiosis, yeah.
It's tricky because it's like I was thinking because
Yeah, because it's different, because we produce, as human beings, our sex cells through meiosis. Plants do it a little bit differently. So it's the sporophyte which has to undergo that spore-producing process, which involves meiosis. Okay. The interesting thing is you have a diploid sporophyte, and it remains attached to the haploid female gametophyte. Okay. So here you have the foot at the top, which is still attaching it to our archegonium. You have a seta, the calyptra is the outside, and you can see spores. And the sporangium is basically that whole structure. But then there's also these structures called elaters, which look kind of like little lines on the inside. Okay, so here are those elaters again. So what's going to happen is that you have these mature archegonial heads, and the sporophytes actually grow down from that umbrella. So isn't that weird? So if you recall, the mosses, they produce the structure upright, right? But these guys, they kind of dangle down, and then the spores are released here. Okay, so here's the whole life cycle. So make sure that you can go through all these steps through basically fertilization of the archegonium, the growing zygote and embryo. You have meiosis producing the spores in the mature sporophyte. The archegonial heads start to grow those sporophytes downward, and then those spores are produced. Those spores are haploid, therefore when they germinate, they become gametophytes, and they start to produce those rhizoid or root-like structures. And then you have archegoniophores and antheridiоphores, which are again those holding structures that hold the different male and female gamete producing structures. So when a raindrop hits the antheridial head, hopefully that there's an archegonia close by. Okay, sometimes you'll get a whole series of them that may be female and a whole series that may be male, and there's no female around, right? So how do they reproduce in that case? Huh?
it's kind of sad. It's like,
They can always clone themselves, right? Through those little tiny gemma or baby plantlets. Okay. So here are the antheridiоphores. So they're really kind of cute. They look like this little flat umbrella. So here's another one. We're looking at male, and this is a phallus liverwort. Notice how the females look different. Okay, so you have archegoniophores bearing archegonia. And remember, those archegonia are sort of on the inside of the under of the umbrella. But notice what's kind of growing here. See this growing down? Those are the little tiny sporophytes. Aren't they cute? Okay, so here you have another female with the sporophyte growing down. And here you have a female where basically the sporophyte hasn't happened yet, right? Because we can see those archegonia, they're still kind of thin structures. So basically, prior to fertilization and after fertilization, everything happens on that archegoniophore, okay? It's not like the mosses where you have just the tips of those phyllids where everything's happening. So here you see the sporophytes. And typically it has three parts: the foot, seta, calyptra, and spores. So the calyptra is the outer covering of that. And at the very top of the foot where it attaches, there's actually a placenta. So what do you think the purpose of the placenta might be? Yeah.
nourish the embryo.
Yeah, to nourish the embryo. So this whole idea of a placenta is not new. Okay, they also have hyaline seta. Okay, so what in the world is that? So it's basically a very thin, translucent, and ephemeral structure. So it doesn't last very long. So here you see the haploid spores and the diploid elaters, which actually help with the movement of those spores to the outside. And here's an up-close look at some gemmicups. They look like little teacups. And again, here's some real examples. So A, B, and C. So A is asexual, right? B, is that an archegoniophore or an anthoridiophore? Yeah, so anthoridiophore. And then C, by default, yeah, archegoniophore. And here's a golden crested newt on a liverwort.
We're learning about it too.
They just really like to be on liverworts. It's like one of their favorite habitats. Okay. So let's talk now about division Anthosaurophyta, the hornworts. Okay, so these are a little bit different. They also have a thallus gametophyte, so it's kind of flat. But the sporophyte lacks a seta. It's only a foot and a capsule. Drinking coffee. All right, the stomata are present in the sporophyte. Okay, isn't that interesting? So why might that sporophyte need to have stomata? Mark?
Gas exchange and moisture exchange so it doesn't like rot.
Yeah, so let's think about what happens when that sporophyte grows. The further it grows away from that gametophyte, the more prone it is going to be for water loss. So this is one of the first places we ever see stomata. And it was just an adaptation to water loss. And gas exchange is important too. So, you know, a lot of these guys, they do some gas exchange, but it's kind of impeded in some cases by living in water. And they also have, bless you, they also have mutualistic relationships with end-fixing cyanobacteria. So often you'll see, if you were to take a hornwort and maybe soak it in water, you would actually see some green algae, it's not really green algae, cyanobacteria, which is like a photosynthetic bacteria. But people often refer to it as algae. So this is kind of neat. The mature sporangium, it basically splits. You kind of untwist in order to release those spores. On here you can see cyanobacteria on the epidermis. So this is a thallus. And check it out. What's that?
Stomata.
Yeah. So it looks pretty similar, right, to the guard cells we've talked about. So here you have a mature sporangium splitting. You can see the stoma right there on the right side, and then we have a tetrad of spores. So tetrad meaning a group of 4, right? So if it's 4 cells together, what cellular process is that usually indicative of? Meiosis, right? Because mitosis is only 1 division. Meiosis is 2 divisions, and you end up with 4 cells. So if you have a difficult time remembering the different processes, just remember that tetrad of spores, that sporophyte actually underwent meiosis in order to produce those spores. And then there's something called the central strand, which basically holds a lot of those together. And it also facilitates the splitting of that sporophyte. So they're derived from a fertilized egg. So each one is from a different egg. And the bottom is haploid. Okay. So you have vertical linear sporophytes that produce the haploid spores. And the flat leafy haploid gametophyte is going to produce both eggs and sperm. So they're kind of weird. So their structures for sperm and egg production are not quite as prominent as we've seen in the other plants so far. Okay, so an egg and sperm, they're going to unite to make a zygote. And then the zygote undergoes mitosis, and it's going to make that growing sporophyte. Okay, so there's a sporangium, and that sporangium splits, and it releases haploid spores. And then the spores disperse, and they're going to undergo mitosis to produce a new gametophyte. Okay, so what the heck is going on at the foot? So obviously this thing is different, right? So here you have the spores on the top, and a later, some stoma, and epidermis. And then you have something called a spore mother cell. And the spore mother cell is going to be involved in the meiotic processes that produce those spores. And then there's something called the meristem in the foot. So the meristem obviously is going to be responsible for growth and differentiation of tissues, just like we saw in the other plants when we were talking about processes. But this is one of the first places that they actually call it a meristem. So here we can see a real slide. You can see the meristematic region. How can we tell by looking at this that this is the meristematic region? Someone other than Mark, yes.
Small cells.
Yeah, so small cells, what else? What's going on in those cells? Yeah, you can see division. You can see a lot of cells in different parts of the division. And then you have the foot and the placenta at the very bottom. Okay, so here you have a real picture of a gametophyte and sporophyte foot. And you can see the sporophytes and the gametophytes there. So they're small. So this one is a different species than the one I brought in. And this one's a bit leafier. Here's another close-up view of them. This is what they look like after they've released their spores. Okay. So here's another one. This one has a very thick sporophyte that grows off in comparison to the others. And here are some other ones coming off. And you can see that foot pretty well, right? The foot's kind of extending above its thallus or leafy structure. So we call it dehiscence. So whenever something is releasing seeds or spores, we call that dehiscence. So this one's dehiscing. Okay, so now we're going to move on to the mosses. So the moss that I brought in is in the process of making the sporophyte. It has not yet quite gotten to producing the sporangium on the top, which gives it its golf club look. So here's a close-up view. So the leafy part, sporophyte or gametophyte? Gametophyte. Sporophyte. Okay, so here you have some moss-covered forest soil. When the weather gets a little bit better, I want to take us down to the St. Francis Watershed Trail because there's some really cute button mosses. I call them tribble mosses because they look like tribbles from Star Trek. Yeah, it's pretty awesome.
You were going to say they look like Dr. Trimble.
I haven't cited them.
Oh, yeah, they don't look like Dr. Trimble. Yeah, you had a question, Mark?
So like, what do you define good weather as? Like when it's like 10 degrees, 5 degrees? I'm talking Celsius, of course.
When it's not too muddy, that's what I'm going for. Because you guys will have to haul me out of there. Because I'm still not so great on uneven ground. I'm still recovering from back surgery, and it's very frustrating as an ecologist to be like really hurting on soil. But anyway, we have a really great moss diversity, and a couple of my students are working with me this semester to build a living wall. And most people, have you guys ever seen living walls? No. Okay, they're really neat. It's basically like a planter that you put up against the wall, and there's plants that kind of come down the sides, and they're very beautiful. But we're going to try to do one with mosses and maybe some lycophidans of some kind, because they're actually endangered. But most people don't care.
They have their ones.
Yeah, and I don't understand why they don't care. Okay, so they all have these leafy gametophytes. What's the ploidy level? Diploid or haploid? Haploid. Are these true leaves?
No.
No. So they do not have a central vein. They do not have real vascular tissue. So here's another really cool moss. It's very similar to the one I brought in. And this one, congratulations, it's a protonema. So protonema, this is after a moss spore germinates, essentially. And when it germinates, it produces these stringy structures, which are kind of like, they're not quite rhizoids yet, but they're going to produce rhizoids, and on the top, we can see a bud going out to the left, and that's going to grow up into that leafy gametophyte. So in one of my little terrariums, I have some protonema that's forming on the top. Okay, so let's look at our mosses. So they have a spore that is dispersed and germinates. It grows these protonema with a bud, so we saw the protonema bud. And then the rhizoid grows off, and we have the young gametophytes, the male and the female. Is there really that much of a visible structural difference between the male and the female? No, you'd have to really look at it under a microscope. Okay, so when you look at it up close, what you'll see is a very similar structure. We have that kind of neck and cup structure. Okay, so the sperm are going to enter in this way. Okay, then we have the antheridial heads. We have these antheridia with sperm on the inside. And those sperm actually have two flagella. So when you look at plant sperm under the microscope, it doesn't move in a very organized manner. Okay, so the embryo then grows and it remains attached. So wherever you see one of these coming off, you know that it had to be a female, right? Because the female is going to support the growth of that embryo. I'm doing it backwards again. Okay, so sperm comes in, fertilizes the egg. This is the venter. The zygote grows into an embryo, and then we have that young sporophyte. Sorry about that.
It's fine.
Okay, so here you have the moss sporophyte, and we have the capsule at the very top. The capsule is going to be having those spores in them. It has a calyptra, which is actually top covering. In order for that to release the spores, it has to come off, essentially. I think. All right. So there's two synapomorphies. There's something called the operculum and the peristome teeth. So the operculum is another word for that top covering, and peristome, that means around the mouth. Okay, and it has these little teeth that are going to release those spores. So here we can see the popping off of the peristome and the release of the spores. All right. So I think I'll have Seth watch this on his own, because I don't know how this is going to record, so I'm going to pause it here. Okay, so just a reminder for Seth and others absent today, to see the life cycle videos, what you need to do is just click on this link right here and it'll take you to the video. All right, so I'm just going to show you some pictures of different mosses so that you can recognize the different structures. So we have our sporophytes are coming off of the female gametophytes because the sperm fertilized the female gametophytes. Here's an archegonial head of a moss called Minium. Here we can see some more moss archegonia. So the eggs are produced there, and this is where fertilization occurs. So the sperm travels down that neck, right? How did that sperm know that it should have gone into that neck? Yeah.
Sperm attracted.
Yeah, so
My
there's actually
question is,
sperm attractant, yeah.
How does the archegonia know to release the sperm attracted? Because like
I
you
don't
only
know.
get so much, right?
I don't know. That's just one of those things. Remember we saw The Secret Life of Plants, and we discovered that they're weirder than we thought they were. And what were these parentheses for?
To keep the water in.
Yeah, so it's just like anything where you increase the surface volume or surface area to volume ratio, what you're gonna get is, it's going to hold on to things better. So here's some archegonia. Here you can see some flagellated sperm. Okay, here's an antheridial head in the moss gammatangia. Some more moss sporophytes. And we can see a moist capsule versus a drying capsule. So remember that this peristome with the teeth, as it dries out, the teeth open to release those spores. And here's a really close-up view. So what's happening there? Okay, remember that the sperm is going to fertilize that female archegonium and then grow into the sporophyte. So they're dispersed by the wind. What do they develop into? What does the little moss spore grow into initially? It's the gametophyte, but the very first protonema, right? So protonema, and then the sporogenous tissue here is diploid, but it undergoes meiosis to produce what?
Spores.
Yeah, so it produces spores. Okay. The interesting thing though is that the sporophyte absolutely depends on the gametophyte for everything. So water, nutrients, everything. So here we can see some spores. So the little tiny dots there. And you can see it's covered here, the capsule, by something known as the calyptra. So if the calyptra is haploid, we know that that was a part of the gametophyte. Remember how it popped off when the stalk started to grow? So a few general features. They have leafy gametophytes, protonema. They have wiry and persistent seta. So you can kind of play with those seta a lot and not hurt them too much. And then that calyptra is going to protect the capsule until the spores are ready. And we have the mouth with the peristome teeth that open up. So an interesting thing is that if you have red sporangia on this particular species that only grows on moose dung, so we're talking a really specialized moss, right? So it only grows there, and it is going to attract flies, okay? And what's interesting is that the flies are dispersing the spores for that moss. So why might a fly be a really good dispersal agent for dung? Someone other than Mark. What happens when someone just leaves, like an animal poops outside or something? What does it attract? Yeah, so this is a really good adaptation because the flies are attracted to the dung and they're going to take those spores and move it to another moose pile. This is kind of weird. So here's another view of it. So they sometimes call it umbrella or petticoat moss. So it's very rare. So all of these again are the bryophytes in general, and we can see kind of the distinguishing features here. So the hepatophytes have oil bodies and hyaline seta. We have the Anthosauropida, the hornworts, no seta. It's mutualistic with cyanobacteria. So this one's really weird. And then you have the bryophytes with all of these different things. and it has that capsule with peristome teeth and the persistent calyptra. So if you see those structures, you know that you're looking at groups of organisms in this particular group. All right, so I'm going to stop here today.